The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hello children, how do you do? Hope you are healthy and happy. Dear children, just as water lily or the lotus flower which grow in water and comes up through water and does not get damaged, in the same manner we should grow up in this world by practicing peace, love and kindness, and we should not get damaged or become tainted in this world. By saying this, I move on to today's session. Today we are moving to the second part of chapter number 8, that is mineral nutrition, and in that part we are going to understand about the metabolism of nitrogen. In the metabolism of nitrogen, we are going to understand about what is nitrogen cycle. The movement of nitrogen from the atmosphere to the living organism and back to the atmosphere is called nitrogen cycle. What is nitrogen cycle? It is a movement of nitrogen from the atmosphere to the organism and back to the atmosphere, that is called nitrogen cycle. And next one is nitrogen fixation. The process of conversion of atmospheric nitrogen into inorganic nitrogenous compound, that is ammonia or nitrogen oxides, is called nitrogen fixation. What is nitrogen fixation? It is a process of conversion of atmospheric nitrogen. That nitrogen which is present in the atmosphere is being converted through this process into inorganic nitrogenous compounds such as ammonia or nitrogen oxide, and that process is called nitrogen fixation. This is again important. Now, nitrogen fixation is done by two methods. Nitrogen fixation which has been taking place there, it has been done by two methods. First method is physico-chemical nitrogen fixation. The second method is industrial fixation. The first method, we can say there electrical nitrogen fixation is taking place, where conversion of nitrogen into nitrogen oxide by lightning and ultraviolet radiation is taking place. With the help of lightning and ultraviolet radiation, the conversion of nitrogen is taking place into nitrogen oxide. This is the natural process through which the atmospheric nitrogen is being fixed there in the soil. And number 2 is industrial fixation. In the case of industrial fixation, we can say it is also a source of atmospheric nitrogen oxide. Example, forest fire, industrial combustion, etc. Through forest fire, industrial combustion, the atmospheric nitrogen is being fixed there in the form of nitrogen oxide. So, this is the first method, that is physico-chemical nitrogen fixation. In that, two methods are there through which the natural process is taking place for the fixation of atmospheric nitrogen into the soil. Now, the second method is biological nitrogen fixation. From the word biological, we can say living organisms are there which are helping for the fixation of nitrogen in the soil. Here what is happening? Reduction of nitrogen into ammonia by living organism is taking place. That is, the nitrogen is been reduced to ammonia by using living organism, and that is called biological nitrogen fixation. It is again important. In the case of biological nitrogen fixation, we can see ammonification, nitrification and denitrification. Three methods are there in the biological nitrogen fixation. Number 1 is ammonification, number 2 is nitrification, and number 3 is denitrification. The first one is ammonification, decomposition of organic nitrogen into ammonia. The organic nitrogen which is present there, it will be decomposed in the form of ammonia, and that process is called ammonification. What is nitrification? It is the conversion of ammonia into nitrate. When the ammonia is formed there, when it is converted to nitrate, that process is called nitrification. You can see here nitrification is involved in two oxidation process. Nitrification is taking place by two oxidation processes. Number 1, the ammonia produced by ammonification is an energy rich substrate for nitrosomonas bacteria, and they are oxidized into nitrite. So, the first step, in the first step what is happening, the ammonia which is formed through the process called ammonification, that ammonia is oxidized into nitrite first. This is nitrite. So, in the presence of oxygen, ammonia is getting oxidized to nitrite, and that is in the second step what is happening, the nitrite is getting oxidized to form nitrate. So, these are the two processes which is involved in nitrification method, and this is very important because in the question paper it used to come by asking the enzyme which is helping or activating this process. In the first step what is happening, ammonia is oxidized in the presence of nitrosomonas to nitrite, and in the second process the nitrite is getting oxidized to nitrate in the presence of nitrobacter. So, you have to study in this manner, my dear children, because in the question paper this question used to come, nitrification process used to come, and you have to answer to it, and you can answer if you are understanding the two steps related to nitrification. So, that is related to nitrification. Now, the third one is denitrification. It is a chemical process in which nitrate is reduced to molecular nitrogen. The nitrate which is produced there through nitrification is again converted back to molecular nitrogen. That process is called as denitrification. So, through ammonification what happened? The molecular nitrogen is converted to ammonia. Through nitrification what happened? The ammonia which is formed there is converted to nitrate by two steps, and the third process is denitrification, where again the nitrate is being converted to molecular nitrogen. Now, you can see here what is happening there, isn't it? This is ammonia, first nitrification, this is ammonification, and then nitrification is taking place, and after nitrification, that is from ammonia nitrite is formed there, from nitrate is formed there, and this nitrate again is giving rise to the molecular nitrogen, and this process is taking place there. Okay, you can see here nitrification process where the microorganisms which are participating in this process is nitrosomonas and nitrobacter, and second one is denitrification, where again the enzyme, or we can say the microorganisms, are participating there in converting the nitrate to ammonia, and those organisms are Pseudomonas, Acromobacter and Micrococcus. Pseudomonas, Acromobacter and Micrococcus are the active microbes which are participating in the conversion of nitrate to nitrogen. I think you would have understood about the three methods related to ammonification, nitrification and denitrification. This is very important, my dear children. You have to understand and study and learn about the active microbes which are participating in these processes. Now, the next one is biological nitrogen fixation. We are going to understand in deep related to this fixation. Here it has been said reduction of nitrogen to ammonia by living organism. The first step it has been said that there is reduction of nitrogen is taking place, and it is taking place with the help of living organism. The nitrogen is being reduced to ammonia, and there are biological nitrogen fixers which are doing this process, and they have the capacity to convert atmospheric nitrogen to usable nitrogen compound. The atmospheric nitrogen, or we can say the nitrogen present in the atmosphere, will be fixed by these microorganisms in the soil in the form of usable nitrogen compound, so that the plant will be able to absorb these nitrogen compound for their growth and development. These nitrogen fixers are prokaryotes, and the nitrogen fixers which are fixing the nitrogen to the soil, they are prokaryotes, and they are free living or symbiotic. We know what is symbiotic or symbiosis, isn't it? So, these prokaryotes, they are free living or they are symbiotic in nature. Again, it is important. And the free living nitrogen fixing bacteria are aerobic bacteria, they are Azotobacter, Beechernicia and Bacillus, whereas the free living another bacteria are there, and they are anaerobic bacteria, and they are Rhodospirillum. And the other free living cyanobacteria are present there, and they are Anabina nostoc, isn't it? These examples are again important, my dear children, because in the question paper the question used to come based on the example asked related to the free living cyanobacteria or the free living anaerobic bacteria or the free living aerobic bacteria. So, you have to understand and study the examples of free living nitrogen fixing bacteria which are helping to fix nitrogen into the soil. Anabina always used to come in the question paper, isn't it? As they are cyanobacteria. Next is symbiotic nitrogen fixation. What do you mean by symbiotic nitrogen fixation? We can see nitrogen fixation when it is carried out by symbiotic microorganisms like bacteria and cyanobacteria is called symbiotic nitrogen fixation. So, what is symbiotic nitrogen fixation? It is the fixation of nitrogen with the help of symbiotic microorganisms, that is bacteria or cyanobacteria, that is called, or that process is called symbiotic nitrogen fixation. Now, the best example for the symbiotic nitrogen fixation is rhizobium. Rhizobium is a bacteria which is the best known example for fixing the nitrogen into the soil, and this rhizobium, it inhabits in the root nodules of legume such as alfalfa, sweet pea as well as garden pea. In the pulses we can see rhizobium, and it is seen in the nodules of these plants. In the root nodules of these plants we can see the bacteria is living there and helping to fix nitrogen into the soil, so that these plants will be able to do the nitrogen fixation process very easily. This example is important. Alfalfa and rhizobium is helping for fixing nitrogen in this plant. Now, next one again, second example is again very important. The microbe Frankia produces nitrogen fixing nodules on the roots of known leguminous plants such as Alnus. So, Alnus is to Frankia, Rhizobium is to alfalfa. Alfalfa is a plant which is leguminous, which is having leg hemoglobin as a pigment in them. But Alnus is a non-leguminous plant. That means these are the plant where leg hemoglobin pigment is not present in them. Is still Frankia, the microorganism, is helping for fixing nitrogen in such plant. So Frankia is to Alnus, Rhizobium is to alfalfa. Next one, Rhizobium and Frankia are free living symbiont and can fix nitrogen. So Rhizobium and Frankia, these are the microorganisms which are able to fix nitrogen. They are free living and they are having symbiotic association with the root nodules of the pulse plant. I think you would have understood about the nitrogen fixing bacteria. Now we should understand how the root nodules are formed, how the nodules in the roots are formed. And what are nodules? Nodules means they are bulb-like structures seen in the roots of leguminous plants. Nodules are bulb-like structures seen in the roots of leguminous plants. We can see how these bulb-like structure or nodules are formed in the roots of leguminous plants. You can see formation of root nodules involves a sequence of multiple interaction between rhizobium and roots of host plant, and the stages involved in the formation of nodules are: number 1, rhizobia multiply and colonize the surrounding of roots and get attached to the epidermal and root hair cells. Rhizobia, first what it will do, it will start to divide itself, it will be multiplying by itself and it is forming a colony around the epidermal cells as well as the root hair cells. In the root hair cells, near to the root hair cells, it is forming a colony there by multiplying by itself. That is the first stage of nodule formation. In the second stage, the root hairs curl and the bacteria invade the root hair. As the bacteria are surrounding this root hair, that moment a chemical interaction is taking place between the bacteria and the root hair cells. That is, the bacteria is producing a chemical, and when it produces a chemical from the root hair cells, a chemical is produced there, and in between them there is a conversation. The chemicals which are produced from both the side is having an interaction, and because of that root hair which is there will be getting curled, and because of that what happens, the bacteria will be getting inside the root. An infection thread is produced carrying the bacteria into the cortex of the root, and in this manner infection thread is formed there and making the bacteria to get inside the cortex of the root. You can see here the bacteria is forming a colony over here. This is root hair, isn't it? And the bacteria, they are forming colony near to the root hair, and in the second stage what happened, you can see the root hair curled, a hook like structure is formed there, and this is the bacteria, and later on the infection, this is infection thread. Okay, this is infection thread which is formed there, which is making the bacteria to enter through it into the cortex of the root hair cell. This is the cortex of the root hair cell, and into it the bacteria is moving. And next one, then the bacteria are released from the thread into the cell, which lead to the differentiation of specialized nitrogen fixing cell. So where the bacteria are getting, and again where they are making or they are getting together, that is, it has been said that bacteria is released from the thread into the cell and they are differentiated, and a tissue is formed there itself. In the cortex area a tissue is formed. It is a specialized tissue where the numerous bacteria are present there, and the nodule thus formed establishes a direct vascular connection with the host for the exchange of nutrient. So, in this manner, that tissue which is being getting differentiated and getting specialized there is forming a structure, and that is a bulb like structure, and that bulb like structure is the nodule formation. In this manner, a nodule is formed there. You can see here, till here we have seen, isn't it, the bacteria has entered there, and this is the specialized structure formed there. So, here a specialized structure is formed there, and this specialized structure is the nodule formation, or we can say the nodule, where we can see the bacteria, they are colonizing themselves and getting multiplied themselves, and thus they will be fixing nitrogen into the soil, so that the leguminous plant will be able to take the nitrogen in the another form, and they will be able to use it for their growth and development. So, this is the right diagrammatic representation of nodule formation. It is very, very important, my dear children. In the first step, bacteria is getting colonized. The second step, an infection thread is formed there, where hook like structure you can see. Through this infection thread, the bacteria is entering into the cortex, and there it is forming a specialized tissue, and that specialized tissue is being in the form of a bulb like structure, and that bulb like structure is the nodule formation. I think you would have understood about how the nodule is formed there. You can see the next point there related to nodule formation. Root nodules act as the site for symbiotic nitrogen fixation. It contains the biochemical components, that is enzyme nitrogenase as well as a pigment like hemoglobin. So, the root nodule which is formed there by the help of the bacteria rhizobium is acting as a site for symbiotic nitrogen fixation. There nitrogen fixation is taking place, and in that area biochemical components are present there, which is used or which are needed there for the fixation of nitrogen, and those components are number 1 enzyme nitrogenase, and number 2 a pigment like hemoglobin is present there. So, these two biochemical components are present there in the root nodules of leguminous plants, which are helping for fixing the atmospheric nitrogen. Now, the first one is being said as enzyme nitrogenase. We can see what it is. Nitrogenase, it is the N2 fixing enzyme, that is nitrogen fixing enzyme. Number 2, it is a molybdenum ion protein. So, what is nitrogenase enzyme? It is a molybdenum ion protein. It is a protein in which molybdenum and iron is the component present there, and it catalyzes the conversion of atmospheric nitrogen to ammonia. So, what it is doing? It is helping to convert atmospheric nitrogen to ammonia. That is the function of nitrogenase enzyme. Now, we can see this enzyme is highly sensitive to molecular oxygen. It is highly sensitive to molecular oxygen. That means, if the concentration of oxygen is high, it will be binding with oxygen, isn't it? So, here we can see what is happening there. The enzyme nitrogenase binds to the molecule nitrogen gas. This is nitrogen gas and this is enzyme nitrogenase, and it is binding to form enzyme substrate complex. This is enzyme substrate complex, where this is substrate and this is the enzyme. And now what is happening there? Reduction reaction is taking place. The hydrogen molecule is getting combined with the nitrogen, and thus the bond between the nitrogen is getting broken there, and the hydrogen molecule will be combining there with the nitrogen, and like this in the second step also reduction process is taking place. Again we can see one more bond has broken and two hydrogen, again it has been joined with nitrogen, and in the last step again you can see the complete bond is been broken there, and the 3 nitrogen, that is, this is the formation of ammonia. So, in this manner the atmospheric nitrogen is being converted to ammonia with the help of enzyme nitrogenase, and later on this enzyme nitrogenase will go back for the second cycle, where it will be combining with the other nitrogen gas for the formation of ammonia, and the product you can see ammonia is formed here. Okay, so this is how the enzyme nitrogenase is working for the conversion of the atmospheric nitrogen to the simple, or we can say to the another form of nitrogen, that is ammonia. So here you can see the enzyme nitrogenase binds a molecule of nitrogen gas, and next one, the hydrogen atom is getting pairing with the nitrogen, and in the third case final product, two molecules of ammonia are formed there, and nitrogenase enzyme is released for binding with the another nitrogen. This is how the enzyme nitrogenase is working for the symbiotic nitrogen fixation. Now this is important, my dear children, you have to understand it, it is given there in your textbook. Now the second biochemical component related to symbiotic nitrogen fixation is leg hemoglobin. It is the pigment found in the root nodules, and this pigment gives pink color to the root nodules. Leg hemoglobin is a pigment and it is giving pink color to the root nodules. And the function of leg hemoglobin, nitrogenase enzyme is highly sensitive to oxygen, therefore leg hemoglobin has the ability to bind with oxygen. So what will happen? So it keeps oxygen away from nitrogenase enzyme. So as nitrogenase enzyme is needed there for conversion of atmospheric nitrogen to ammonia, and it is very sensitive for oxygen. If oxygen is coming there and if it is in higher concentration, surely this enzyme will be binding with oxygen and the fixation will not take place. Therefore, leg hemoglobin, what it will do? It is having the affinity or ability to bind with oxygen. So this hemoglobin will bind with oxygen and keeping the oxygen away from nitrogenase enzyme. So we can say that due to this character, leg hemoglobin is an oxygen scavenger in the root nodules. This is again important. So leg hemoglobin is playing the role of oxygen scavenger. It is protecting the oxygen from the nitrogenase enzyme. Therefore, it is known as oxygen scavenger in the root nodules. I think you would have understood about the oxygen scavenger, that is leg hemoglobin in the case of root nodules. Now next one we can see the conversion of nitrogen to ammonia needs energy. The nitrogen, atmospheric nitrogen, which has to be converted to ammonia, it needs energy, that is 8 ATP for each ammonia. For the production of each ammonia, 8 ATP is needed there, and this 8 ATP is obtained from the respiration of the host cell. In the host cell, as we know, these cells, they are respiring. So during respiration, ATP is produced there, and these ATP are utilized there for the production of ammonia. In this manner, symbiotic association of nitrogen fixation is taking place in the plants. Now you can see here biochemistry of nitrogen fixation. Nitrogenase complex is formed there, and it is a key enzyme in nitrogen fixation. The overall reaction related to is, you can see here, nitrogen plus 8 hydrogen ion plus 8, that is 8 proton and 8 electron plus 16 ATP is needed there for the production of 2 ammonium ion, where the conversion of this ATP is leading to 16 ADP plus 16 inorganic phosphate. So this is the overall reaction which is taking place there in the symbiotic nitrogen fixation. It is again important, my dear children, you have to just go through it, as nitrogenase complex is very important, and with that overall reaction related to symbiotic nitrogen fixation is also important. You should know from where this ATP came for the symbiotic nitrogen fixation. I think you would have understood about it, my dear children, very nicely. Now, next one is fate of ammonia. The ammonia which is formed there is not directly absorbed by the plant. It has to be converted to its some other form. Then only the ammonia will be taken up by the plant for its growth and development. The other form is it will be synthesized, or we can say synthesis of amino acid. Ammonia is converted to amino acid, and amino acid has an amino group and a carboxyl group. In the case of amino acid, it is having an amino group as well as a carboxyl, or we can say an acid group. The synthesis of amino acid takes place in two ways. In two ways the amino acid is been synthesized. First one is reductive amination and second one is transamination. Reductive amination, in this process ammonia reacts with alpha ketoglutarate acid to form glutamic acid. Ammonia, it will be reacting with alpha ketoglutarate acid to form glutamic acid. Okay, my dear children, that is the first part of how amino acid is being synthesized. You can see here reductive amination. Alpha ketoglutarate acid when combining with ammonia in the presence of the enzyme glutamate dehydrogenase, this is important. And here NADPH is converted to NAD. So, this reduced compound is getting reduced to form NAD, and in the presence of glutamate dehydrogenase, this is converted to glutamate, or we can say glutamic acid. Now, the second one is transamination. A new amino acid is formed by the transfer of amino group from one amino acid to the keto group of keto acid. So, transaminase, from the name itself we can say transfer of one amino group to the keto group is taking place there, that is transamination, and this reaction is taking place in the presence of transaminase enzyme. Here you can see first one, what is happening there. Amino acid is being transported or is being transferred in the presence of transaminase to keto group, or we can say as alpha keto acid, and this alpha keto acid is being again converted to amino acid. So, in this manner this reaction is reversible reaction where amino acid is been transferred to keto acid and keto acid is again transferred to amino acid. This process is transamination reaction. So, with the help of these two reaction, the synthesis of amino acid is taking place, and in this manner amino acid is being taken up by the plant for its growth and development. Now, you would have understood about the fate of ammonia, my dear children. As ammonia is toxic in nature, it will not be directly absorbed by the plants. So, it will be converted to its nearest form, and among those nearest form, one form is the synthesis of amino acid and the other form is in the form of ammonium ion. Okay. So, synthesis of amino acid is taking place by two processes. One is reductive amination and second one is transamination. Both these processes are very important. It used to come in the question paper, my dear children. You have to understand and study it. It is there in your textbook. Then last but not the least topic, that is amides. These are the derivatives of amino acid in which the hydroxyl group, that is OH, is replaced by amino group, that is NH2. So, what are amides? These are the derivatives of amino acid where hydroxyl group is replaced by amino group. These are transported through xylem vessels. And two important amides which are found in plants are asparagine and glutamine. Asparagine, they are formed from aspartate, that is aspartic acid, and glutamine is formed from the glutamate, that is glutamic acid. So, from aspartic acid, asparagine, which is an amide, is formed there, and glutamic acid, or from glutamate, glutamin, which is an amide, it is formed there. So, in this manner, two important amides we can see in the plants are asparagine and glutamin. My dear children, I think you would have understood about it. By this, the chapter is completed here, and you have to go through the second part of this chapter very nicely. It is very important. You have to understand it, you have to learn it. It is very easy chapter, my dear children. Just go through it 1, 2 or 3 times. You will be understanding it very clearly, and take down the notes. If you are having any doubt related to any topic from this chapter, you can call me and clarify your doubt. Please, dear children, don't keep your work pending. It can cause or it can give you negative consequences. So, my dear children, please be always on track, be positive, be happy, be healthy. By saying this, I wind up the session here. In the next session, we will be meeting again. Till then, take care.